Bismillah ar-Rahman ar-Rahim. In the name of Allah, the most merciful, the most kind. We again now take up paper 6.1, which is going to be now in the new syllabus. It is going to be called paper 4. So don't think paper 4 is anything else. It's an alternative to practical. That's why it's called an ATP, alternative to practical. Now, as you all know, alternative to practical, this is November 22, paper 6.1, and this is going to be for 1 hour. So everybody please be very clear, this is going to be a 1 hour paper. Now, as you all know, to answer all the questions, the spaces provided, there's no choice in here. And this is what everybody has to know, there's no choice, you have to do the entire paper. Foods can be tested to determine the types of molecules that they contain. A student decided to carry out 3 tests to investigate the composition of 2 foods, A and B. A. He then cut that cube into 3 pieces, each of the same size. 1 piece was used for each of the 3 tests. So what did we have? First we had a big cube, 1 centimeter, 1 centimeter, 1 centimeter. They cut the cube into 3 pieces. So we had 3 pieces like this, each of the same size. Well, this doesn't look the same size. So let's make it a little better. So we have a cube, and then we have, let's make a cube. Might not be very good at drawing. So cube, and then you cut it into 3 pieces. So well, I'd say one here, and one here. So 1, 2, and 3. So 3 pieces. 1 piece was used for each of the 3 tests. He carried out the same procedure with B. For the first test, he followed these instructions. Label a test tube A1. Label a test tube A1. Cut one of the 3 pieces of A into small pieces and put the small pieces. So you took this piece, this top piece off, and you cut it into small pieces and you put it into A1 and put the small pieces into the test tube. Pour 2 centimeters of ethanol, and then we poured 2 centimeters of ethanol, 2 centimeter cube of ethanol. Ethanol is not blue, it is a clear solution. So we added ethanol to it. Shake the test tube well. Place the test tube in a test tube rack to allow the contents to settle at the bottom. He repeated this procedure with one of the pieces of B with the test tube labeled B1 and placed it in the test tube rack. State what you would use to produce the 1 centimeter, 1 centimeter, 1 centimeter cubes of A and B. What would you use to cut something? Well, something very basic. You can't say a knife. You'll have to say scalpel or a razor blade. Even a knife was around, and a ruler, because you had to cut it. So you had to have a scalpel, and you had to have scalpel and a ruler. You could have said a knife, you could have said a razor blade, but the ruler was the one which was important, because in order to do this sizing, you had to have the ruler. And I'm sure a large number of students would have missed out the ruler part, because you won't think of the ruler. Because I told you, you have to think as if you're doing this in the lab. And if you think you're doing it in the lab, only then you would think that you needed a ruler for it, because you had to cut it a certain dimensions, certain length, 1 centimeter by 1 centimeter by 1 centimeter. Part 2 of the question, describe what you would do to ensure that the test with the small pieces of A and B were comparable. Yeah, this 1 mark. So same number of pieces, or the similar number of or similar size or similar surface area of the small pieces, and the same time for shaking. Because you see, this was given in the above procedure. You can see that it said you have to shake it. So shaking means you should have, actually you even get electronic shakers, so which will do it at a certain speed. Shake the beaker, you just put the beaker on it and then they vibrate and they will shake the contents of the beaker. So describe what you would do to ensure that tests with the small pieces of A and B were comparable. So that means comparable means they must be the same situation. So same number of the small pieces and the same time for shaking. So same number of small pieces and the same time for shaking. But you only have to write one of these, not both of these, to get 1 mark. The student poured equal volumes of distilled water into 2 new test tubes labeled A1W and B1W. He took test tube A1 and carefully poured the liquid into test tube A1W. He followed the same procedure with the test tube B1, pouring the liquid into the test tube B1W. He observed the mixture and recorded his observation in his notebook, right? So equal volumes of distilled water into two new test tubes, A1W, B1W. Then test tube A1 carefully poured into the liquid in test tube A1W, and then followed the same procedure with B1, and then recorded the observations in his notebook. For the second test, he used the second piece of A and B cut into small pieces. The small pieces of A were added to test tube A2 and the small pieces of B were added to test tube B2. He added the same volume of distilled water to both test tubes. He then used a glass rod to stir the contents of both test tubes. Explain why he cleaned the glass rod after using it in test tube A2 and before using it test tube B2. You see, why did we clean the rod after using in test tube A? Okay, so now if you use a rod and you have stirred the mixture in A2, well, you don't know which molecules are present in that food. Maybe it has a reducing sugar, maybe it has a fat, maybe it has a protein. Now, some of that will stick to the glass rod. And then you use the same glass rod again in this, and that has those molecules sticking to it. So this test would also become this. Maybe this food does not have that molecule. So explain why and avoid cross contamination. Or you could have used some other word. Cross contamination is not a word that you, so that you know to not transfer the contents of A2 to B2, because maybe there are some molecules in A2 which are not present in B2, but when you're using the same glass rod, well, you're going to contaminate it. So this word cross contamination is of course a very, very high level of English. I'm sure we who do not speak the language, not our mother tongue English, we would find it difficult. But I would say we can just say so as not to transfer or not to contaminate or to not transfer the molecules in A2 to B2. Next, he then added the same volume of biuret reagent to both test tubes. The photograph shows a graduated measuring cylinder with the biuret reagent that he added in one of the test tubes. State the biuret volume, the volume of the biuret reagent. Volume. So volume here is what? You can see here. What is the volume? 7 centimeter cube. So writing 7 got you a mark. It was a very easy mark. I hope everybody got that. After adding the biuret reagent, he observed the mixtures in test tube A2 and B2 and recorded the observations in his notebook. I mean, very good student, observes everything and puts it in his notebook. I haven't not seen many students like that. For the third test, he placed the third piece of A and B on a white tile, added some iodine solution, and recorded his observations in his notebook. The student's notebook is shown. Wow. Appearance of solutions after test. Test tube A1W colorless. Test tube B1W cloudy. Now, this was the ethanol story. So you know the ethanol emulsion test is for fats, and when it is clear, no fat, when it is cloudy, fat is present. Test tube A2 and B2 were pale purple and darker purple. So this was the biuret reagent. This is the protein test. So this darker purple would mean more protein, and the pale purple would mean less protein. Iodine was brown, so no starch, and was iodine on B was also brown, so no starch. Now this is my assumptions which I am just talking to you, and then let's see what they have to say in the table. Complete the table by using only the information from his experimental method and his notebook. Test number 1, 2, 3, test solution, food A, food B, and this is for 5 marks. Wow, it's a lot of marks. Now you have to just complete the table using only the information from the experimental method and his notebook. Now there are 5 marks for this. What are the marks for? All 9 spaces completed. So you got a mark. And you wrote appearance or you wrote observations. You wrote appearance or you wrote observations. I didn't say you write both of them. I said only one of them. And then you wrote ethanol, biuret, and iodine solution. You got a mark for that. And then you placed all of these in the correct line. You did it correctly. So you got a mark for that. So all you had to do was just copy it down from the previous thing. This thing had to be copied down here, not the conclusions. Just it said complete the table using only the information from his experimental method and his notebook. his or her notebook, whatever it was, who was a his or a her. So what we have to understand is that we were only asked to do that. And that is why I tell you to read the whole question, because sometimes you would have written the conclusions in the previous table, and then that would have been, then you would have to cut it out and then rewrite it, because remember, you can't use the eraser here in Cambridge exams. So use the students' observations to state what you conclude about the composition of foods of A and B. So test one, what did you conclude? And look at this, there's 6 marks. So for every, you've got to remember there would be 2 points. That's why there's 6 marks. This is what comes to your mind immediately, that is, you look at the marks and then you say to yourself, okay, so what have I to write here for test one? For test one, all you had to write was, it was a fat test. If you didn't know the word fat and you had written lipid, well, that was fine as well. Lipid was correct as well. So you got 1 mark for that. And then what was it? What was the test? What was the result? Yes, it was present in B but not in A. So present in B but not in A. Right? Now test 2 was the biuret test. And what you conclude about the composition? Use the students' observations to state what you conclude about the composition of foods A and B. So the biuret test was, of course, the one that you had done, but you don't have to give the biuret. What was it tested for? It was tested for protein. The ethanol test was for fat and lipid, which we talked about earlier. The biuret test was for protein, and it was purple, but it was present in both. So it was, what was it? Let's look at it again. It was pale purple and dark purple. So that means this had less protein, and dark purple means it had more protein. But even then, if you didn't know this and you just wrote present in both A and B, protein in both A and B, you got a mark for that. But of course, there would be some brilliant student who would know that the shade of the purple was giving you more. So more protein in B, more in B than A, more protein. So you got 4 marks. Then test 3 was the one in which you use the iodine. And what do we use iodine for? It's called the starch test. So the starch was not present in A or B because it remained brown in both of them, not present in A or B. Or you could have said no starch. So this got you your 1 mark, 2 marks, 3 marks, 4 marks, 5 marks, and 6 marks. So I mean, you can get full marks in this. It's like maths. If you get the answer right, you get all the marks for it. Next part of the question, he found that it was easy to cut A into a cube and divide it into 3 equal parts. However, they found that B crumbled easily, fell apart, which made it difficult to cut to produce a cube and to assure that the 3 parts were of equal size. Suggest how the problem could have affected the results in test tube A2 and B2 and your conclusions. So that's 3 marks. So how would you have ensured how this problem could have affected the results in test tube A2 and B2 and your conclusions? Well, because there would be different surface areas for A and B, and there would be different volume or amount for A and B, and we could not have concluded that there's going to be more protein in B. So how would we have more protein in B results? So were not very comparable, because if it was just becoming very powdery, or it was breaking up into tiny, tiny pieces, and then how would you decide, okay, that you were putting it all into equal pieces? So suggest a change to the method to help overcome this problem. Well, it's easy. You use the same mass. So you use the same mass of A and B instead of cutting it into pieces. Well, you just measure the mass. Say it was 2 grams, so it would be very easy to measure 1 gram each. So this was something which they expected you to come up. This was a difficult 4 marks, but then, as I tell you, the main thing is that you think you're doing this in the lab, and how would you sort of conclude about it? That how would you do this? Is measuring length and all is not a very technically scientific a good way. Measuring the mass is more important. So in A and B, of course, different surface areas for A and B, and then different volume for A and B, and cannot conclude more protein in B, and results were not comparable. A change in the method or overcoming or improvement in the method was weighing the mass of A and B rather than cutting it up into pieces. Question 2. The photomicrograph shows a section through a small artery in a muscle. Magnification is 90 times. Always look at it very carefully. It says artery. In the space below, make a large drawing of the artery. Do not draw individual cells. Make a large drawing of the artery. So all you had to do was give me a sharp pencil, continuous outer line, no shading, oval shape, size at least 80 millimeter, and only the artery drawn, folded inner layer, and outer muscle layer thinner than the inner muscle layer. So the outer muscle layer was thinner than the inner muscle layer. Let's try and draw it. Now the diagram that you had to draw was only this thing. That's all what you had to draw. You have to just make a large drawing of the artery. Do not draw individual cells. So I always tell you, I do this in A levels as well. There's a lot of, we do this, it's called a plan drawing. Well, you just sort of you just trace it. And you see there is this line which is changing. There is this muscle layer. Of course, you don't know it, but we'll teach you in A levels. And there was this line that was changing. So this thing, and then you draw this. And this is, of course, what you all have to do. So there were 4 marks with a sharp pencil, continuous lines, continuous outer line, no shading. The important thing is no shading. Please do not shade this. Oval shape, size at least 80 millimeters. So draw a big diagram. At least 80 means it should be not less than 80, it can be more than 80. And only the artery drawn. Folded inner layer. This was the folded inner layer. This was the folded inner layer. And the outer muscle layer thinner than the inner muscle layer. So this is the outer muscle layer. This was thinner than so the outer muscle layer was thinner than the inner one. So this one was the one which was, so this one was thinner as compared to this, the inner one, right? So this got you your 4 marks. Then coming to the B part of the question, draw a straight line on the photomicrograph to join lines C and D, measure the length of this line. So what you had to do is you had to draw a straight line on the photomicrograph. So to join lines C and D. So this is the line that I've drawn, C and D. And then what I do to measure the length of this line. Now this was 68. What? The 3 marks. 68 millimeter. Calculate the actual width of the artery and give your answer to 2 decimal places. So you have to divide 68 by what? The magnification was 90 times. So you have to divide it by 90. 68 divided by 90. This got you 1 more mark. So 68 millimeter, 1 mark, 68 by 90, another mark, and 0.76 millimeter. 2 decimal places. So 0.76 millimeter. And this would get you your 3 out of 3. As the heart pumps, it pushes blood through arteries, causing them to expand and contract in response to the flow of blood. Each expansion and contraction is a pulse. Describe in detail how you determine your pulse rate when at rest without using an electronic monitoring device. Well, that was easy. What? Sit quietly for a few minutes. Use your finger at the wrist, and then you count it for 30 seconds. Count the pulse for 30 seconds, and then you calculate it per minute, and you do several counts, and then you calculate the mean. Because you might get a little excited, and the pulse might go up. So this is how we always do this, is we always tell the person to sit. As a doctor, I always tell the patient to sit quietly for a little while, and then we count his pulse, and then we count it again if it's a little high, because the person can get a little nervous, more adrenaline output, and the pulse rate can go high. So sit quietly for a few minutes, use the finger at the wrist, count for 30 seconds and calculate the value per minute, and then several counts and calculate a mean. This paper had a question 3 as well. So, a tree produces fruits that are attached to the thin leaf-like structures called bracts. One fruit with a bract is shown in the diagram. Magnification is 1 time. Usually the whole structure shown in the diagram drops from the tree. Sometimes only the small fruit drops without the bract. A student investigated how far fruits were dispersed when they fell from the tree. An outline of the tree is shown in the diagram. So we have a tree, then we have 10 meter, and then the outline is shown. She used a 1 millimeter into 1 millimeter square frame at increasing distances from the base of the tree, and she placed this frame on the ground to look at any fruits inside the frame. This is called a quadrat. Some fruits still had bracts attached and some did not. She counted the numbers of each and recorded them. So you see, they fell from the tree. Some fell here, some fell here, some fell here. So they fell at different places around the tree. Some here, some here. So use a quadrat or use a frame and at increasing distances from the base. So we place the frame here, then place the frame here, then place the frame here, and so on and so forth all around the tree. So some fruits still had bracts and some did not. She counted the numbers of each and recorded them. Her results are shown in the table. Distance from the base of the tree in meter is 0, 5, 10, 15, 20, 30. Number of fruits with bract 15, 23, 16, 8, 4, 2. Number of fruits without bracts 15, 35, 38, 0, 0, 0, 0. Construct the line graphs of this data on the same axis on the grid below. So a line graph of this data. So we have actually now in this situation, we have two datas. One is this and one is this. And how many do we have? We have 1, 2, 3, 4, 5, 6. So we have 6 here, 6 here. So we have 12 points that we have to plot. And the distance is 0 to 30. And as I told you in many of my time and time again, I tell you in each paper is this will go on the x-axis. This is the first column. And this is the independent variable. And this will go on the x-axis. The first column is the independent variable and will go on the x-axis. Now this is how I've taken the scale because I wanted 30 at the end here and I wanted 40 at the top here. So this is roughly how I've taken the scale, and I've written this because you see the labeling of the x-axis was the most important. So distance on x-axis and axis fully labeled with units, you got 1 mark for that. Then continuous linear scale for both axes and over half of the grid used. So we're using more than half of the grid and values at origin. At 12 points plotted correctly and straight lines correctly drawn between the plotted points and both lines labeled. So we had to give a key for this. That is why they have 5 marks for this. So there would be a key which I would give for it, and this is how we would do it. I'm just going to plot it for you and then you can see it. As you can see, I just wrote them here on the side and I plotted them. And now all you have to do is you have to connect them point to point with a ruler, straight lines correctly drawn between plotted points and both lines labeled. So I've of course used two different colors, but of course you would have to use either you can use the dot for one and then the other one you can use the cross, or you can just label it on the graph as well. So you've labeled it and of course you get another mark for that. So please everybody have a look at this and see that how the graph has to be done. Please everybody must not lose any marks in the graph. Now, using your graph and the information given, describe the distribution of fruits without bracts. Now, without bracts, of course, these were the ones, the green ones. Now, they're all less than 10. They're all less than 10 meter. So, all under less than 10 meter from the tree or within 0 to 5 meter. You could even said that because they're only between 0 to 5 meter, and then from 10 there is 0, 0, 0, 0, 0, 0, 0. And then the distribution of fruits with bracts. Now, how were they different? Yes, they were different. They were more near the tree, increases to 5 meter and then reduce further away. Range of fruit distribution 0 to 30 meter from the tree. And that you can see from the graph. That was very evident from the graph. So all less than 10 meter from the tree, and then the distribution of fruits with bracts were more near the tree and fewer further away. Range of fruit distribution was 0 to 30 meters. Then part C, state one environmental variable that could affect how far a fruit with a bract is dispersed. Of course, wind. So these are carried by wind, or you could have said air movement. More the wind, more the further they would have been carried away if there was wind. If there was no wind, it was still air, then of course it doesn't affect it. Last is suggest one other variable that could affect how far a fruit with a bract is dispersed. Explain how this variable would affect the distance dispersed. The size, the surface area of the bract. Larger the surface area was carried further away. Then the mass of the fruit. Heavier the fruit carried less far. So the variable and the explanation, you have to give me that. That is for 2 marks. Either you said surface area, so larger the surface area will be carried further away, or you said the mass. So the lighter it is carried away, or you said heavier the fruit will be carried less far. Surface area of the bract and larger the surface area would be carried further away. Or you could have said, so either you said this or you said this. Fruit mass and heavier the fruit would be carried less far. Of course, the heavier the fruit will just land very near the tree. It's not going to be carried far away if it's going to be a heavy fruit. So this is all for the 11 marks, and that finishes this paper. And wish you all the best. Please do this paper before you look at this video. So everybody must attempt this paper on their own and then look at this video. It's no fun in looking at this video if you're just going to read the mark scheme. No, first you do it yourself and then you look at all my explanations which I've given you. Please attempt the paper in writing before you watch this video so that you can learn from your mistakes. If you have written something and then you look at this video and you realize, oh my God, this is how I was supposed to think. So don't just watch this video without ever doing the paper yourself. First, you must attempt the paper, do it in writing, and then look at this video. And then, of course, you will learn a lot. But if you just don't do this, if you just look at the video and say, okay, well, that's not going to help you much. Best of luck, and please do subscribe and do leave some comments so that I know how all this is helping you. Thank you very much.